everyone. I'm here to talk about my PhD research, my PhD was actually awarded just last month, and it was funded by the Psoriasis Association, so thank you so much for funding it. The aim of it was to investigate the early changes in the skin during treatment of psoriasis. And before I start, I just want to say thank you to all the patients who are involved and the team members at St. John's Institute of Dermatology. And just to give a bit of background, the skin, it's a complex organ with multiple cell types. And in normal conditions, the skin and immune cells communicate, but just like any relationship, it can go wrong. And when the conversation gets disrupted, there is redness, scaling, and in some cases, psoriasis. Now, we're in a pretty fortunate situation where in some cases we have some really effective psoriasis treatments we've heard about today. So biologics are injectable drugs which target specific immune proteins in the body. And as we've heard, they're not available for everyone, but they are currently used to treat severe psoriasis. But there are drawbacks to these biologics. So they require regular injections and they require regular hospital visits for monitoring. It's also the case that they're not a complete cure, so it does mean that they are lifelong treatments, and with any treatment, there is a risk of adverse events. So the aim of this research was to better understand how these drugs actually resolve psoriasis, what we wanted to look at specifically were the very early stages of psoriasis resolution, and the whole point of this was that so by understanding better the mechanisms of these treatments, we can potentially lead to better, newer treatments and also inform on personalized medicine approaches. So how did we actually do this? Well, we decided to look at skin samples just before treatment, 3 days after treatment, and 14 days after treatment after biologic injection. And at this point, I just want to really highlight how important it was that we had people with psoriasis who volunteered their time and skin samples for this research. Honestly, thank you so much. This wouldn't be possible without you. What this allowed us to do was to track the early changes in the skin at a cellular level before visible improvement. And how did we do this? We used a new technology which has come out in the last decade called single-cell RNA sequencing. And what you can see here, this is an infographic that we usually show at our scientific conferences because it's an improvement on an older technology called bulk RNA sequencing. So in this analogy, if we were to look at this fruit bowl, if that's our skin sample and we want to see what's going on inside it, what we used to do was just mash it all up and then basically treat it like a smoothie and just see the general changes in the colors over time. But what we can do now is we can pick out all those individual fruits and actually look exactly what's going on. So that's what we were able to do. We were able to look at the activity of each individual cell in the skin during the early stages of treatment, and this is really new. This was done for the first time. But this is super, super valuable. I was trying to think of ways to convey how much information we actually managed to get from this. And if you think about it, this is over 36,000 genes in over 160,000 cells. That's 6 billion data points. And if you were to print that out onto A4 paper, that'd weigh about 6 tons of the weight of an elephant, and it would take about 20 Arnold Schwarzeneggers to deadlift. Pretty impressive. So what did we get out from all of this information? Well, we discovered a new cell type in the skin, which is in psoriasis inflamed disease. And you can see it here. This is in a separate experiment where we showed where it actually was in the skin. And what we found was that it was found in the upper layer of the skin. And this was done in a separate cohort of people who had volunteered their skin, and again, would not be possible without our volunteers. This gave us new insights into psoriasis resolution. This cell type that we found is a type of fibroblast, and it gives inflammatory signals towards keratinocytes. We proved this in a lab in a separate experiment, and we also showed that in the very early stages of treatment, it's reduced in abundance. So already by day 14, we showed that it was lower, and then we showed in skin that's not affected by psoriasis, it's not there. So why does this matter? Well, it improves our understanding of the complex skin cell types contributing to psoriasis. And by doing this, we get new clues into what's actually going on. And hopefully, as we amass more and more data, because discoveries like this will hopefully inform on the design of better, longer-lasting treatments. Now, this would not be possible without a huge amount of people. There's a real community at St. John's and St. Guy's and Thomas's Hospital, some of whom I've managed to catch up with today, which has been really nice. So we involved in this, there have been doctors, nurses, project managers, technicians, database managers, and scientists, and most importantly, you people with psoriasis who have very kindly donated your skin and samples for which hopefully we can develop better treatments for you. So with that, I just want to say thank you. You make discoveries like this possible. And if you'd like to join future research at St. John's, then please visit this website. And again, thank you so much to the Psoriasis Association, as well as King's College London and NHS and St. John's. Thanks so much.